In this video, we'll talk about the structure of the bacteria, of a typical bacteria. As you know that bacteria have some main structures and other extra structures. The main structures in bacteria are the structures that all types of bacteria should have it, like what we have, like the cell wall, cell membrane or the plasma membrane, and the cytoplasm. And what we can see in the cytoplasm is the DNA and ribosome. So these are the main structures that all bacteria should have it. Hence, sometimes we have other extra structures like capsule, like pili or pilus, like flagella, and outer membrane. These extra structures, some bacteria have it while other bacteria don't have it. The main structures in bacteria, as we said, that we have, the first one is the cell wall. The cell wall here is made up of a layer of peptidoglycan. The peptidoglycan is made up of protein and carbohydrate. And this structure is found in this type of bacteria, which is named as eubacteria, but it's absent in archaebacteria. Now, the second one is the plasma membrane or the cell membrane. The cell membrane or the plasma membrane, it's made up of lipid bilayer, and it's the site for both photosynthesis and cellular respiration. Why both photosynthesis and cellular respiration happen here in the line of the cell membrane? Because they don't have chloroplast and they don't have mitochondria. After that, we have the cytoplasm, which is a viscous solution, and this viscous solution contains the DNA and the ribosome. The DNA is circular and it's a closed loop, and also we have the ribosome. These are the main structures of bacteria that all types of bacteria should have it. The extra structures in bacteria that some types of bacteria have it while other types don't have it are, the first one we have capsule. Capsule is the outer cover that surround the bacteria and protect it against the harsh environmental condition, and also it protects the bacteria against our white blood cell. Then we have also the pili or pilus. The pili or pilus, they are the short hair-like structure. These short hair-like structure that are found on the surface of bacteria, they help in the attachment or adhering of the bacteria on the surface of the host cell, and also it helps in the transferring of the genetic material or the DNA from one bacteria to another bacteria. This pili is made up of protein, while the capsule is made up of polysaccharide. The other extra structure that we have is the flagella. The flagella, it's used for movement, and it's this long hair-like structure and also made up of protein, but it's used for movement of the bacteria. And some bacteria have one flagella, other bacteria have group or tuft of flagella in one side, or maybe in two sides, or maybe the flagella surrounding all the entire bacteria. While other types of bacteria, they don't have this flagella for movement. So how they move? They move either by producing a slime and then they glide on it like myxobacteria, or they move by corkscrew-like rotation like the spiral-shaped bacteria. After that, we have another extra structure. This extra structure is named as outer membrane. The outer membrane, you can find it only in gram-negative bacteria. We can't find it in gram-positive bacteria. And this outer membrane, it's an outer cover that surrounds the bacteria and help to protect the bacteria against the antibiotic. Sometimes the bacteria can have another extra structure, which is named as endospore. Endospore, it's the cover that surrounds the DNA during the harsh environmental condition. For example, during dryness, during when the high temperature exposed to the bacteria, if it's exposed to radiation or chemicals, at that time they surround their DNA by a cover which is named as endospore. This endospore, or the bacteria that have endospore, it's not in a reproductive state, which means the bacteria here have no ability to reproduce, but it's only a protection state, which means only they protect themselves from the harsh environmental condition, and when the environment becomes favorable for them, at that time this endospore will be break down and the DNA start to reproduce, the bacteria start to reproduce, and the DNA to divide again by binary fission.